Tantor Audio, a division of Recorded Books, presents The Jewel Box, How Moths Illuminate Nature's Hidden Rules, by Tim Blackburn. Narrated by Mack. Introduction. The Moth Trap. Nature is painting for us day after day pictures of infinite beauty, if we only have the eyes to see them. John Ruskin. In July of 2018, for my 52nd birthday, my wife bought me a black plastic box, 2 plexiglass sheets, and a light fixture with a 20-watt fluorescent bulb. The box came flat-packed, which meant that I had to draw on my limited practical skills to intersect the tabs with their corresponding slots, resulting in an open-topped cube about 50 centimeters on each vertex. I screwed the electrics to a bar that ran across the top, setting the socket above the box, but sheltered under a wide white plastic disc. The light bulb bayoneted into the socket and hung down. The perspex sheets slotted at a 45-degree angle from the rim, forming a wide transparent slide down into the interior, ending at an opening the size and shape of a letterbox. The black plastic box with a light on top might seem like an odd choice for a birthday present, but it was what I wanted. It sounds dourly functional, but it is also a box of enchantment, one that can conjure life out of thin air. I put it outside that July evening, as dusk was beginning to fall, plugged it in, and watched the glow from the light start slowly to build. Then I went to bed, already excited, hoping the conjuring trick would work. I woke early and expectantly the next morning, and went outside to see. The box had indeed performed its magic, and there inside was the reveal, a scattering of jewels. A moth trap festooned with moths. I'd first got into moth trapping while leading undergraduate field courses to the Kindrogan FSC Field Studies Council Centre in Scotland, now sadly closed to such trips. As a university academic, one of the joys of my job is that I get to interact with young people eager to learn. Yet it's not only student minds that come away from field courses changed. They can be revelatory experiences for teacher and taught alike. Even students who are interested in biodiversity aren't nearly close to knowing all the different forms of life in which they share their surroundings. We always set pitfall traps, plastic cups sunk into the soil like golf holes, to catch ground-dwelling invertebrates. And every year I have to encourage the students not just to pick through the harvest with the naked eye, but also to use a dissecting microscope and to turn the magnification up high. Without this, they wouldn't see the tiny springtails that inevitably fall in. These animals are in the order Collembola, and are among the closest relatives of insects. Indeed, when I was a student, they were considered to be insects. Taxonomic orthodoxy has changed since then, and now they tend to be classified as the earliest divergent branch from the part of the tree of life that is the subphylum Hexapoda, of which insects are the most familiar manifestation. Like insects, springtails have 3 body sections: head, thorax, abdomen, with 6 legs attached to the thorax. Unlike insects, their mouth parts are hidden inside the head capsule. Most springtails have an organ known as the furcula, folded underneath the abdomen, which is responsible for their name. When triggered, this sprung fork can catapult the springtail into the air and away from its enemies. The springtails caught at Kindrogan pack all of this structure into an animal barely a couple of millimeters, 1/10 of an inch in length. This miniaturization, combined with their unassuming habit of hiding in the leaf litter and soil, means that most students don't know that these organisms even exist. The feeling of offering someone the revelation of an animal they'd never even dreamt of never gets old. There was a moth trap at Kindrogan, and one summer I asked their staff to run it. This one was a Robinson trap, a large bucket-type container with a ring-shaped lid and a powerful mercury vapor bulb. Moths are attracted to its light and drop into the container below to await identification and release in the morning. The trap was sited in a purpose-built shed on a grassy bank, looking east over the River Ardell. On that first June morning, the students and I were torn between crowding in to examine what the trap had caught and hanging back for fear of crushing the delicate insects underfoot. Many moths are lured to a moth trap, but don't quite make it inside. Most species sport the colors of camouflage, and it takes some time to train one's eye to spot them in long grass. For some moths, greens predominate for a background of summer leaves. Others are mainly brown for wood and stone. The lemon yellow of a brimstone moth blends surprisingly well into darker tones of grass, especially given the brown patches that dot the leading edges of its wings to break up its shape. Clouded, bordered brindles have a similar strategy to disrupt their outline, and their shades of brown render them shadows between the stems. Even the monochromes of the clouded border are hard to spot among sparkling drops of dew. The shed itself was dotted with insects too. The long white wings of a male ghost moth were aligned with the grain of a board. A coxcomb prominent sat in a gap between walls and roof, looking like a sycamore key that had become lodged as it fell. A gold swift stood out boldly against the grey planking. And then we opened the trap. A cornucopia inside. Buff tips, hawks, carpets, waves, and more. I couldn't identify them all. Not then. But I could see what a proliferation of species we'd summoned. The trap had given us all a glimpse of unexpected biodiversity in a country I thought I knew well. Arriving back at King's Cross after a week out with a group of students is always a time of mixed feelings. Home is just a handful of tube stops away, with a weekend of much-needed rest, sleep, and family time awaiting. Fully immersive teaching with long days in the field is exhausting. But being back in London is always accompanied by an ineffable feeling of loss. After a week spent in the pine forest and mountains surrounding Kindrogan, the city is an impossible accretion of brick and people, of air and noise pollution. Senses that have come alive in the Highlands have to be dialed back down to cope with the urban environment. The journey from Scotland to London takes one through decades of biodiversity loss in a single day, and feeling that loss takes its toll. If you live and work in a city, it's easy to feel detached from nature, but it's still there if you look. Hampstead Heath is a short walk from my London flat, and the diversity of life it supports is surprising. It often helps to adopt the viewpoint of a 5-year-old, getting down on hands and knees, face close to the ground. Even the areas manicured for sport are home to more than just grass. Broad-leaved plantain and knotgrass make a living there, lying too low for the blades of the mower. Just a few feet away, where the heath rangers leave the field uncut, the grass grows tall enough to flower, resolving the stems into fescue, cocksfoot, and foxtail. Yellow buttons of creeping and meadow buttercup mix with the white clover. Common vetch drapes itself across the grass, and creeping thistles lie low if not yet flagged by their flowering spikes. Picnics in the long grass here need care. And of course the plants themselves are al fresco dining for a variety of animals. When it isn't raining, butterflies transect the field, and bumblebees and honeybees make beelines from flower to flower. Caterpillars and grubs chew on the leaves, burrow their way into grass stems and thistle buds, and even mine their way between leaf surfaces. Look up and you may see a bird of prey overhead. A kestrel frequently hovers over the slope here. But common buzzard, red kite, sparrowhawk, peregrine, and hobby all hunt over the heath. An early morning visit might produce a red fox trotting away to cover. More likely mammal sightings are brown rats, or their arboreal cousins, the grey squirrels. Even a short walk can be hard to snatch, though, against a backdrop of work and family. I missed those opportunities for escape. It was something I was feeling especially keenly at the end of that last trip to King Drogon, when the obvious hit me. I could make nature come to me. Why wait a year to run a moth trap again? My birthday trap was a lightweight Skinner, entry-level, with an actinic bulb as light source, running off mains electricity. It was sited in our only outside space, 30 feet off the ground, on the roof terrace of our flat in the London borough of Camden. The terrace overlooks some mature gardens, facing a line of tall limes, with large cherry and pear trees in sight. Yet this is very much an urban location, in an area with no shortage of other light sources. Before that first morning, I wondered whether this restricted patch of green would house any species of moths, and if it did, whether any would find their way into the trap. Happily, the answer to both questions was yes. Experience in science tells us that answers to questions simply lead to more questions. As the sphere of knowledge expands, so does the area of interface between known and unknown. So it was, too, with the moth trap. The most pressing new question that first morning was, what were the identities of all the species that had appeared in the box overnight? This was not a trivial task. There were more than 80 moths to pick out and identify. Giving things names matters. It's how we begin to quantify our experience of the natural world. The moths themselves aren't knowledge yet. The first step is to resolve them into their constituent species. As we will discover, knowledge of most groups of species in most parts of the world is decidedly poor. We're just beginning to describe the diversity of organisms with which we share the planet. Yet there are some notable exceptions, one of which is British moths. Britain is blessed with some excellent field guides to these insects, beautifully and expertly illustrated with paintings and photographs that depict almost all the species found here. Even with these guides, though, it takes time to get one's eye in. Different species prefer different habitats and fly at different times of year, and these ecological details are not apparent simply from looking at pictures in a book. The field guides have this information, but there are hundreds of pages of text to leaf through. It's a slow process. Fortunately again, the UK moth trapper is blessed with additional support. Type "What's Flying Tonight" into an internet search engine, and it will link you to a web page that uses your location and the date to produce a list of the species most often recorded there and then. Each comes with photographs and a calendar bar showing when in the year the adults are on the wing. It's based on millions of moth records logged by the charity Butterfly Conservation through their National Moth Recording Scheme. A wonderful application of accumulated scientific data to the public appreciation of biodiversity. Still stumped? One can go to social media, where experts are happy to help you identify all sorts of animals from photographs. Moth novices can ask at Moth ID UK for assistance. But please consider a contribution for this service if you can. Even with all this help, though, some moths cannot be specifically named without dissecting their genitals, and must simply be logged as part of an AG, to denote that they belong to an aggregate of species that are indistinguishable without the skills of the specialist. I am obsessive about identifying animals, but, so far, I draw the line at killing them to satisfy my obsession. That first morning on my terrace, I began slowly to match species to their names. The Dunbar and knotgrass, tree lichen beauty, gypsy, Jersey tiger, pale mottled willow, and dingy footman, and, most aptly, the uncertain. Most of the moths started with this label, but only 2 finished with it. Adults of this species are very similar to others that fly at the same time of year. More of this anon. Finally, after a good chunk of the morning, I'd assigned 82 individual moths to 28 different species. All these animals had appeared as if by magic on a small roof terrace in urban London. The entomologist and writer E.O. Wilson, who coined the term biophilia to describe the innate affinity people have with the natural world, noted that every kid has a bug period. I never grew out of mine. That morning, I grew back into mine. We've given names to more than 1 million different animal species, but this is certainly only a fraction of the total. Estimates of global animal species numbers range from 3 million to 100 million, depending on the method used to extrapolate from those species that are currently known to science. But the actual number seems most likely to be nearer the lower end of this range. One credible recent study calculates just shy of 8 million species. This is a phenomenal diversity when, as far as we know, the presence of even one living species sets our planet apart from all others. Yet of all animal species so far named, roughly 1 in 10 is a moth. Around 140,000 species in the order Lepidoptera. 1 in 9, really, another 20,000 or so, Lepidoptera, are butterflies, which we distinguish colloquially, but which are just a subgroup of moths that have taken to flying by day. The true number of moth species worldwide is likely to be far higher. Most species are found in tropical forests, which remain poorly explored in comparison to the temperate latitudes in which most scientists and taxonomists live and work. Why, then, given all this diversity, had my trap pulled 28 species of moth out of the London undergrowth? What was it that determined 28? Was there anything that could be done to increase that number? And what would happen if we tried to bring more species into the local environment? Would I end up with a richer catch? Or would present incumbents simply be squeezed out? What about if we took some species away? Would other species move in? Or would our local moth community just be the poorer? There were 28 species in my trap, but 82 individual moths. Almost a third of the catch comprised of 2 species. The tree lichen beauty was the most numerous, with 13 individuals, but the Jersey tiger was close behind with 12. Add in the Dunbar, 8, ribboned wave, and codling moth, 6 of each, and 5 of the 28 species accounted for more than half of the moth's court. Most species were represented by 1s or 2s. Is that typical? Could the species I caught live elsewhere? Indeed, do they? The two most common species in the trap that morning were ones I'd not seen in Kindrogan, which suggested that something was different between these two locations. The habitats around the trap, Camden and Kindrogan, were obviously quite distinct, but perhaps it was just down to geography. Do we expect to get the same set of moths in the trap if we shift it 700 kilometers or not? And what does it tell us if we do? The different sets of moths in Kindrogan and London could also be just an issue of the time of year. Spring and summer come later to Scotland than to southeast England. So maybe the Kindrogan trap would be full of tree-lichen beauties and Jersey tigers come August. No species is an island, entirely of itself. All animals must consume to survive, so the presence of food is important. Moths are holometabolous insects, which means that they develop through a life cycle of egg, larva, the caterpillar stage, which is split into a variable number of instars, punctuated by shedding of the hard chitinous exoskeleton to create room for expansion of the body and allow onward growth. Pupa, or chrysalis, in which the miraculous transformation from caterpillar to moth takes place, and finally adult. Most consumption is done by the caterpillar. This must start out being able to fit inside a tiny egg, but then accumulate enough raw material to effect the metamorphosis to an adult capable of laying eggs of its own, up to 20,000 in some cases. Most caterpillars are vegetarians, but their tastes vary enormously. The lesser broad-bordered yellow underwing has a mouthful of a name, but the list of plants its caterpillars will eat is much longer, from dead nettles, docks and mayweeds, to sallow and hawthorn and blackthorn. The marbled beauty, on the other hand, develops on lichens, which provide slimmer pickings and do not do well in polluted areas. Both species were on my inner London roof terrace that first morning of trapping. Moths are consumers, but are also often the consumed. They're links in the web of life, parts of the habitats they occupy, and habitats themselves, for communities of predators and parasites. The moth trap can illustrate this. It sometimes attracts wasps, the colloquial yellow jackets, especially in autumn, which can bring tension to the morning's proceedings. Yellow jackets are important predators of other insects, doing a largely unheralded service as pest controllers in gardens and crops. They often buzz in to try their luck with the moths resting around the trap. Other wasps are also drawn to it too. Parasitoids that lay eggs in the bodies of caterpillars, hatching to eat the host alive from the inside. Sometimes moths are both consumers and consumed. Those Dunbars I caught grew up as omnivores, feeding on leaves, but sometimes also on the caterpillars of other moths. What effect do all of these interactions have on the populations of the moths I was catching? Are moth numbers determined by what they eat, or by what eats them? There is an extensive and venerable branch of science especially devoted to these questions. This is the science of ecology, which has been my career for the past 3 decades. I'm deeply interested in questions relating to the abundance, distribution, and richness of species, which I mainly research using information on a group of animals that have always been my first love: birds. My familiarity with this sort of question, and with birds, has certainly not bred contempt, but often it takes the contrast of new experiences for us to suddenly see the familiar in a different light. We take our surroundings for granted. My moth trap had given me a new perspective. Ecology was first defined in its modern form as Ecologie, in its native German, in the 1860s. by the biologist Ernst Haeckel. The etymological roots of ecology combine the ancient Greek words oikos, from which we get eco, and logos, for the principle of order and knowledge. Oikos does not have a single meaning. It refers to the family and the family's property or the house. Why it prefixes economy is clear. For the study of the interactions between organisms and their environment, which is one definition of ecology we might use, eco relates to the third meaning, and we often give it a more personal interpretation. Literally, ecology is the study of our home. The relevance and logic of this to an ecologist is obvious. We are organisms, and our environment matters fundamentally to us. Haeckel's formal definition was the comprehensive science of the relationship of the organisms to the environment. Haeckel was a keen disciple of Charles Darwin, and so it seems appropriate that the definition of ecology has evolved over the years. Although Haeckel identified the essence of the subject, he codified it in a form that was arguably too general and too vague. What isn't ecology under his definition? What exactly is ecology trying to explain? It was not until almost a century later that we got a clear answer to this second question, thanks to the Australian ecologist Herbert Andrew Arthur. He revised the definition of ecology to the scientific study of the distribution and abundance of organisms. The crux is to try to understand where organisms are found, how many are found there, and why. With minor tweaks, Canadian Charles Krebs advocated for the addition of the interactions that determine, for example. This is the definition that most ecologists use today. It identifies why ecology is key to understanding the contents of a moth trap. A moth trap may be a source of wonder for the biophiliac, but it is also an effective scientific tool. The animals that it conjures out of thin air are samples of the wider moth community in the immediate area, or, in some cases, the moths that are passing through it. They are a snapshot, a fragment of the wider panorama. By piecing together snapshots, we can begin to see the bigger picture. For moths, the number of snapshots available is huge, at least in the UK. These islands are home to an extended community of amateur trappers who write up their nightly catch and submit details to regional or national record schemes. Since 1968, a countrywide network of moth traps has been coordinated by the Agricultural Research Station at Rothamsted in Hertfordshire. Historical records that antedate the existence of these schemes can, in some cases, be extracted from old notebooks and added to the picture. The high-resolution image that results means that we can start to pick out details, and individual moth trappers can see where the pixels they provide fit into the broader patterns that emerge. These patterns become the basis for ideas about how the natural world works, which we can then put to the test by further observations, or better, by experimentation. Are we seeing a random set of individual animals of a random set of species thrown together by chance? Or are there rules? And if there are rules, what sort of rules are they? Gradually, our understanding of the world around us improves. But the scale of the task should not be underestimated. The natural world is fiendishly complex. Imagine that you had the technology that would allow you to scan our planet with such detail that you could map the identity and location of every individual animal, plant, fungus, bacteria, archaean, and virus. What sort of picture would this give you? Each of the individuals scanned would belong to a species. On top of the 8 million or so animal species, estimates suggest about a further million other eukaryotes, roughly 30% plants, 60% fungi, and the rest protozoa and
Sample complete. Ready to continue?